ELISA, Protocol Types, Advantages and Applications. The enzyme-linked immunosorbent assay involves coating of an antigen to a solid support such as a membrane or a 96-well microplate. The coating is done using bicarbonate or carbonate coating buffer. Proteins which have not been separated by electrophoresis can be bound to membranes and analyzed with primary and secondary antibodies, as in the immunoblotting procedure. Such analysis are often called dot blots. The more common format is to absorb the antigen to the wells of a 96-well microplate and to use substrates that produce a colored product. The ELISA is suitable for the analysis of large numbers of samples, and most of the procedure can be automated. Steps in ELISA: antigen coding, blocking, incubation with primary and secondary antibodies, developing. Antigen coding. The first step is to bind the antigen to the micro-well plate. The antigen used can be a purified protein, peptide, or a crude protein extract. The choice of antigen will depend on the application and the nature of the antibodies being tested. Binding to 96-well microplates is achieved by incubating the wells with a solution containing the antigen. Protein binding to the plate is due to hydrophobic interactions between the protein and the plastic. Coating incubation may vary from 2 hours at room temperature or overnight at 4 degrees Celsius. Blocking. Once antigen is bound to the plates, it is important to block the remaining surface of the plate or membrane, to prevent nonspecific binding and the detection antibodies during subsequent steps. Many blocking buffers are available to block the free sites on the plate, BSA, non-fat dry milk powder, etc. In PBS or TBS with minute percentage of tween 20 or Triton X100 can be used as blocking buffer. The protein in the blocking solution will attach to the membrane in all places where the target proteins have not attached. Thus when antibody is added, which will bind only to the target protein, so the background interference will be reduced. Incubation with primary and secondary antibodies. After blocking, the excess blocking agent is removed by washing the plate or membrane with PBS and tween 20 for some time. Then the antigen-containing wells are incubated with the primary antibody. Appropriate antibody dilutions need to be made which will better results. Serial dilutions are often carried out, and the minimum titer which is giving a good response can be used. Optimization of the antibody concentration need to be done for getting better results. Antibody dilution are made in PBS or TBS. ELISAs are typically carried out in duplicate or triplicate. A positive and a negative control is also included along with the test samples. Primary antibody incubation may vary from 2 hours or even more. Incubation periods are required to get good results. After incubation with the primary antibody, the wells are washed extensively to remove any unbound antibody. Similarly, membrane or plate is incubated with secondary antibody with suitable dilutions. Incubation period varies from 2 hours or sometimes even more. Secondary antibody will bind to the primary antibody. Secondary antibodies are conjugated with enzyme which on reaction with substrate in the developing solution will yield color. Excess antibodies are washed off with wash buffer. There are variety of tags or enzyme labels can be conjugated to the secondary antibody. Most widely used enzymes are horseradish peroxidase and alkaline phosphatase. Developing. A developing solution having chromogenic substrate is added to the wells. The enzyme reacts with the substrate and gives color. The intensity of the color will be proportional to the amount of secondary antibody, which proportional to the amount of specific primary antibody in the sample. Alternatively, a constant amount of a defined antibody can be used to quantify the amount of a specific protein in the sample. ELISA plate reader is basically a spectrophotometer designed to read the individual wells in a 96-well microplate. ELISA plate readers are interfaced with a computer to assist in data management. Types of ELISA. Competitive ELISA. In competitive ELISA, antigen is incubated with an unlabeled antibody. These bind each other to form antigen-antibody complex. These bound antibody or antigen complexes are then added to an antigen-coated well. The plate is washed, so that unbound antibody is removed. there will be competition for binding of antigen in the well, then a secondary antibody specific to the primary antibody is added. This secondary antibody is coupled to an enzyme. When a substrate is added, enzyme acts on the chromogenic substrate and emits color. ELISA reader is used to measure the intensity. Sandwich ELISA. In sandwich ELISA, first the plate is coated with a capture antibody, to the antibody-coated well sample is added, and if any antigen present in the sample will binds to capture antibody, an antibody which detects the antigen is added, and this antibody binds to antigen. Finally, an enzyme-linked secondary antibody is added, and this secondary antibody binds to detecting antibody. When a substrate is added, the secondary antibody having enzyme converts the substrate and color is developed, which can be read in ELISA reader. Advantages and applications of ELISA. ELISA can be used to quantify antibody in the sample. ELISA can be used to quantify antigen in the sample. Large number of samples can be processed at a time. Highly sensitive. ELISA has many applications in vitro disease diagnostics. Some of them are: detection of HIV antibodies in serum, diagnosing Lyme disease, detecting plant diseases, application in toxicology for screening of drugs, detecting food allergens.